Welcome to the STEM Super Scientist series.
Hello, this is Shema. Today we are diving into the fascinating world of skeletal muscle anatomy and physiology, focusing on two crucial structures, the tubule and the sarcoplasmic reticulum. These organelles play a vital role in initiating and regulating muscle contraction. Skeletal muscle cells are packed with myofibrils, which are surrounded by the sarcoplasmic reticulum. DSR, a specialized smooth endoplasmic reticulum, appears as a blue sleeve around the orange myofibrils in this image. Its primary function is to store calcium using active transport when the muscle cell is at rest. The SR stores calcium and releases it on demand. When the muscle fiber is stimulated to contract, calcium release from the SR provides the final go signal for muscle contraction. The SR tubules run longitudinally along the myofibril and communicate with each other at the H zone. Larger perpendicular cross channels called terminal cisterns are found at the AI band junction, always occurs in pair. The SR is closely associated with large number of mitochondria and glycogen granules, which are involved in producing the energy required for muscle contraction. At each AI band junction, the sarcolemma muscle cell membrane form T-tubules that penetrate deep into the cell. These T-tubules are crucial for rapid transmitting changes in membrane potential into the muscle fiber, facilitating the release of calcium from the SR. Terminal cisterns, also known as insects, are a large area of the sarcoplasmic reticulum that form perpendicular cross channels at the junction of the A band and I band in a muscle fiber. Where the T-tubule meet the SR, we find a structure called a triad. A triad consists of T-tubule flanked by two terminal cisternae of the SR. This proximity is essential for efficient communication between the electrical signal and calcium release. This triad is important for the rapid transmission of electrical impulses from the sarcolemma to the SR, ensuring synchronized muscle contraction. T-tubule conduct electrical impulses from the surface of the muscle fiber deep into the cell. This ensures the signal to contract reach all parts of muscle fiber simultaneously. When an action potential travels along the sarcolemma and down the T-tubule, it triggers the release of calcium ions from the SR into the cytosol, the aqueous components of cytoplasm of a cell. That help to have the uh, excitation, contraction coupling. Calcium release happens when the action potential in the T-tubule causes voltage-sensitive protein to change shape, open calcium release channel in the SR. The influx of calcium into the cytosol initiates the sliding of myofilaments leading to muscle contraction. In this video, we learn that when a nerve impulse arrives at the neuromuscular junction, it triggers an action potential that travels along the sarcolemma. This electrical signal propagates down the T-tubules as the action potential reaches the triad. It triggers the release of calcium ions from the SR into the cytoplasm. This sudden increase in intracellular calcium initiates muscle contraction. Thank you for watching.
That's a wrap for today's episode of STEM Super Scientist Series. Thanks for watching. If you enjoyed this video, don't forget to hit that subscribe button. Until next time, keep learning, stay curious, and we'll see you in the next STEM Super Scientist episode.